really delighted. First of all, everybody knows this is the New Feinberg Institute seminar, and our topic today is donating organs for research, the genotype tissue expression project experience. And I think we're going to learn a tremendous amount, not only about the particular project, but about the issues that surround it. I'm really delighted to introduce, we have two guests today, both from NIH. Sarah Chandros-Hull, who went from Chandros to Chandros-Hull while she was here, I remember that well, is a faculty member in the NIH Clinical Center's Department of Bioethics, just a wonderful bioethics program at NIH, for those of you who don't know about it. She also directs the NHGRI Bioethics Core. For those of you who don't know Sarah, she actually did some of her training here at Hopkins, as in her doctoral degree is from Hopkins and also from the program in health policy and bioethics. So it's wonderful to have Sarah come home, as it were, and give us a talk. Jeff Struewing, Dr. Struewing, is a program director in the Division of Genomic Medicine at NIH at the NHGRI. He has multiple responsibilities, among which he is the program coordinator for the Genotype Tissue Expression Project of the NIH Common Fund that we're going to hear about today. So you came to NIH in 2007, or no, you came to the Office of Population Genomics in 2007?
Yes.
Kind of a lifer NIH. Yeah. Well, it's a great place to work. So we're really delighted to have two colleagues from NIH with us. Please join me in welcoming them to conference.
Thank you very much, everyone. It's a pleasure to be here. Good afternoon. When Sarah and I talked a little bit about coming here, I think our first notion for both of us, we would do this without any slides. And then I think Sarah kind of caved first a little bit. And then I said, okay, I'm going to do two slides, a title slide and an acknowledgement slide, which you always would have to have. Of course, we both end up with sort of full slide deck. So we'll try to make this, I'm going to keep mine under 20 minutes. I think we'll try to keep both of our talks to 15 or 20 minutes and then leave a lot of time for questions, because at least with gtex, invariably there are questions. This is sort of my acknowledgement slide from June. This is a big, complicated project. I put some of the names in red for this one because they're the key people on the whole donor recruitment and sample collection part of gtex. So John Lonsdale and Jeff Thomas from NDRI, Richard Haas and Gary Walters, the two organ procurement organizations, Gift of Life Donor Program and LifeNet Donor Force at NDRI. Laura Siminoff is conducting an ELSI study from Virginia Commonwealth. Barb Foster is the PI at Roswell Park Cancer Institute, one of the other collection sites, and they work with Susan Sullivan and Jason Bridge from the upstate New York Transplant Service, a very sort of high-volume organ donation center. And it's a site that's done many, many research studies associated with the organ donation and tissue donation process as well. And really, the success of gtex so far is due mostly to those names in red, the people that work under them, because it was really a challenge getting this whole thing set up. And I'm simply the coordinator. There's a lot of NIH people that work on this. This is funded out of the NIH Office of the Director. It's a so-called Common Fund project. So this has multiple institutes participating in it. They tend to be big projects that don't really fit necessarily into one institute or whatever. And I have the privilege of coordinating that. So this is our sort of overall goal. It's really to establish a resource, both database and tissue bank, in which to study the relationship between genetic variation and gene expression in reference or non-diseased human tissues. So we take individuals who pass away from any cause. There will be a smattering of chronic diseases. People don't end up in an autopsy suite for no reason. But we're not focused on any disease process. So a lot of these organs will be effectively normal. The total resource will be about 900 post-mortem donors, and we're collecting multiple tissues, a couple dozen, 2 to 3 dozen from each donor. And the idea is to look at the association between your natural genetic variation and gene expression across this wide range of tissues, because every cell in the body has the same genetic blueprint, the same genetic code you got from your mom and dad and all those variants. But a kidney cell and an eye cell are dramatically different, even though they have the same genetic code. If we were to sequence them, they'll have the same sequence. But they're different because of differences in gene expression, developmentally and over time. So different genes are turned on and off in different cell types, and that's why they become the cell types they are. And it's a complex sort of regulatory network. And it probably distinguishes, a big part of the distinguishing factor between us and our closest relatives like primates, because only a few % of our genome actually gets made into proteins. So, less than 2% of all the 3 billion bases gets turned into a protein. All the rest of it is the regulatory switching and rheostating genes up and down. So, and a lot of the discoveries in the last 4 or 5 years, the genetic variants, the loci that we now know are associated, lead to increased risk of breast cancer or of heart disease in this. Most of those map outside of the protein coding region of the genome. Areas that are difficult to interpret. So both from just understanding gene regulatory networks generally, and to give us a bit of a handle on translating some of these findings into things that will help human beings, I think gtex will really help. I think that was mostly what I was going to say about the scientific background. I'll say one more thing sort of near the end, but of course if there are any questions or things that, if the question and answer period, I'll be happy to answer them. But I'm going to try to focus as much on the donor recruitment and sample collection and those issues as possible. Normally when I give this talk, I focus more on the print, and I definitely try to tailor it to these issues. But as I was driving here, I'm thinking of just some other issues that didn't sort of make it to the slides, and hopefully we can discuss later if questions come up about them. So these are all the moving parts of gtex. It's a big, complicated thing. There's a laboratory data analysis and coordinating center that does all the molecular work, the brain bank. We have a funding statistical methods groups that are really diving into this data. We're less than a fourth of the way into it now. We've collected approximately 200 post-mortem donors, so we're just getting started. It was funded as a pilot period because it was concerns and just worries that we would be able to set up such a complicated tissue collection process to yield high-quality RNA. And our focus is gene expression. That comes from RNA, which is a very labile molecule. So, there was enough concern that we didn't get sort of the full funding initially until we showed that we could get the system set up and actually get high-quality RNA. And so, a lot of the lower portion revolves around that. I'm going to focus just a bit more on what's inside this red box. This is all the donor recruitment and tissue collection. It's sort of coordinated and organized by NCI's cancer hubs, CA HUB, Cancer Human Biobank. This is done mostly through their prime contractor, SAIC Frederick. So between SAIC Frederick and NCI, we funded a sort of wet lab, the biorepository operations and pathology processing center. We've got a data coordinating center, so we collect clinical and epidemiologic information from all of the post-mortem donors. And that's associated with the data. We have a very robust pathology review of every single specimen undergoes pathologic review. It gets stained, and a pathologist reads it to see the quality of the tissue and whether it has any disease processes. But then the tissue collections performed by what we call biospecimen source sites. There are 2 subcontracts to National Disease Research Interchange, that's the NDRI, and those are their 2 organ procurement organizations I mentioned, Roswell Park Cancer Institute and Upstate New York Transplant Service. And then the ELSI study is a subcontract of NDRI for the Virginia Commonwealth University. This is a big, complicated project to manage. There's a combination of grants and contracts and soon-to-be cooperative agreements, and there's an intramural component, and the NIH Office of the Director, and all the other institutes. So it's a big, complicated thing, but fortunately, things have gone, I would say, about as well as we could have hoped, mostly due to the professionalism of the people at the biospecimen source sites and their OPO and AATD sort of partners. I mean, so we've really sort of tapped into the organ procurement, organ donation system, because they're ultra professional. They're just very accustomed and comfortable with talking to families at these very, very difficult times, approaching them about donating a solid organ or tissue for human transplantation, and then talking to them about gtex if they're so inclined. I've sort of talked about this a tiny bit. The first bullet, because this is post-mortem research. It's not legally human subjects research, but from the very beginning, we sort of made the decision that we were going to get. We often fall into calling it consent, but it's really authorization from the next of kin. The organ procurement organizations are relatively careful to call it authorization and not consent, because it's really about performing research or procedures on a deceased human being. So we get authorization from the next of kin from every single donor. We make as much of this data freely available as possible. If it's summary data, it doesn't have any direct genetic sort of identifier sort of things. If it's for the genotyping information, for the RNA-seq that has genetic variant information, goes into the controlled access part of dbGaP, even though, again, legally, I guess we could have tried to get around that. gtex is, we often sort of describe it as an addendum to the authorization form that's used for organ and tissue donation. In some sites, it's not exactly an addendum, it's sort of more integrated into it. But basically, it's after the OPO staff go through the process of explaining and seeing whether they can get authorization for organ donation, that then they describe the gtex study. So it's relatively short. It's at the end of what is already a relatively long either telephone interview or in-person interview, because these tissues, especially in a tissue transplant setting, are essentially an elective procedure. They go to great lengths to get a social and behavioral history to rule out any potential infectious diseases that could transmit something. So there's, the families of these donors go through a lot leading up to the potential for organ and tissue donation, and then gtex is added on at the end, and it's relatively short, but it covers a number of things that Sarah will describe in a little bit more detail. Things that we would look for in a typical consent that involve genomic research. We do also have an ELSI study that's led by Laura Siminoff from Virginia Commonwealth. It's both a qualitative and a quantitative study, really to sort of delve into some of the factors that are affecting whether people consent to tissue donation for biobanking or our study. And really with the idea that if we found sort of issues or problems, to really make the process as good as we could going forward. Again, everything was set up for a 2-year pilot. As of last month, I think Laura and her team had completed about 90 interviews of family members, including 25 from families who declined participation in gtex. So I think that this is pretty interesting and sort of unique to have a pretty sizable proportion of people that you're able to talk to and understand why they didn't participate. So we've gotten just little hints of this in our meetings. Again, we're still very early on in this, so there's a lot of people are anxious to get to the results, but really it's sort of, we've been concentrating on getting it into the database and getting our tissues into the biobank and getting things going, and now we're trying to pedal as fast as we can to begin to make access systems for the data and actually begin analyzing it. This is sort of the whole flow of what happens, getting back a tiny bit to the science. But so on the left are the autopsy donors. Again, the size will be around 900. We're collecting a small number of tissues from a small number of donors. This was always a relatively small part of gtex, and it got off to a somewhat late start. We've only enrolled, I think, less than 20 donors, and we're not even sure how much longer we're going to continue that for various reasons. But pretty much all I'm going to talk about today, and most of the discussion relates to the individuals on the left. So from each donor, we on average collect between 25 and 30 different organs or tissues, depending on the gender and whether an organ is transplanted. So anything related to solid organ donation or tissue donation takes precedence. I mean, we don't mess with that sort of process at all. So we don't sample any of those organs that are transplanted. So somebody could be a gtex donor if they donate a solid organ like a kidney or heart, lung. We then don't have those tissues within that suite of tissues for that patient. But so there are about 35 or 36 tissues that are on our list for collection, but depending on various things, it usually is somewhere 27, 28, 29. if we have permission to collect the brain, we got a couple of regions, small little regions in the autopsy suite. And all of these tissue amounts are fairly small. They're sort of eraser head of a pencil sort of size, what would fit in a typical pathology cassette. We get a couple of aliquots from each of these organs. This is a big, complicated process. It takes 1.5 to 2 hours. And of course, everything is labeled and barcoded, but getting everything into the labeled tubes and everything recorded. There's an incredible suite of SOPs and quality management system that NCI and CA Hub set up to make this really sort of industrial strength sort of quality management system for the whole tissue collection. Again, part of it gets put into a paraffin block and reviewed by a pathologist. In the center there, that red box, or leading into that, we get a blood sample from every donor as well. And that's the source of the DNA for the genotype, and we get a very dense SNP genotype, so that's our genetic variation information. We also attempt to establish a lymphoblastoid cell line, so one of the white blood cells within the blood sample, you can treat in a way to try to make it grow forever, or nearly forever. We also send one of the skin aliquots in media, and we attempt to establish a fibroblast cell line. This is another cell line that would grow effectively indefinitely. And the fibroblast cell line would be the source of the IPS cells that Sarah's going to tell you a lot more about. I think I'm already running out of time. I'm just going to skip through any more about that and get sort of to this issue. But, so gtex and the induced pluripotent stem cell, I realized I probably, like grabbing up, I should have put a slide in about what an IPS cell is. So these are, well, the name is induced pluripotent stem cell. So this is taking a fully differentiated human cell and treating it relatively simply to sort of de-differentiate it so that it can be then differentiated into other cell types. You can take this skin thing, treat it so that it becomes a bit more immature-like, and then it can be treated in such a way that it can become a neuron or neuron-like cell, and a muscle or muscle-like cell. And then these grow and can be used in lots of both basic and somewhat more translational research. So we got the fibroblast cell line thinking that's one of the cell lines, one of the cell types that the procedures are worked out for making the IPS cell lines. And so gtex is obviously, well, it's a pretty unique sample collection. And to set up this infrastructure and do this, we have teams that are on 24/7 ready to go to activate when a family gives consent. So it's not likely to be replicated often or very soon. But we get a relatively small amount. We have a few hundred milligrams of tissue typically. So if it gets used up initially, so it's a limiting resource. It's, we're going to make access to the scientific community to that, but it will run out. And so sort of questions like, how do gene regulatory networks that we can study in our post-mortem tissues, in our sort of not totally normal, but in our sort of just straight human tissues, how would that compare to gene regulatory networks from induced pluripotent stem cells? These iPSC cells just have such incredible potential for medical research that having a comparison from the same individual to know what a differentiated renal cell from an IPS line looks like physiologically and genetically and gene regulatory-wise compared to a kidney cell that was taken directly from that organ. gtex if we had both of these, both standard peripheral tissues and IPS lines, it would really be an ideal setting in which to address many basic questions and just give us a bit of a sort of calibrator for things we will learn in IPS cells. I've gone over time already. So these are just some of the URLs that are associated with this. I think, well, the talk or these slides can be available. One that I would point you to, the second to the last, are all the SOPs for this study are published, including essentially the consent forms without the actual site names, and a tiny bit of it sort of obscured, but the consent forms, the MTAs, everything is there. And I'll stop there. I guess plan would be probably to have Sarah speak, and then while Sarah's coming up, everybody has a quick question.
Yes, you mentioned that RNA is very labile, and we know that expression profiles change drastically and almost immediately upon initiation of hypoxic conditions. How did you validate your RNA samples?
That's a great question. I mean, and that's, I mean, so, yeah, I mean, these are post-mortem tissues, so a couple of ways. Well, that was part of the reason for having the surgery tissues, that we would have a handful, like 5 tissues that we would have with no post-mortem interval. They'd have surgical, things. So we'll have that as a comparison. We're comparing a lot to existing surgery gene expression studies, and we've got a small little methodologic study where we're taking some organs and holding them, we're sampling them immediately. And these are ones where we get within an hour or so post-mortem, and then resampling the same organ over time to get, there's definitely going to be differences. Fortunately, what we've looked at so far, when you clustered the kidneys from our post-mortem with sort of surgical or all other kidney specimens, they cluster completely together. So it's always going to be a criticism. I'm sure there's going to be some, and we can see some genes that go way up or down very, very quickly. Hopefully we'll be able to sort of define that signature, if you will. But basically, when you look at it from a gene expression standpoint, just do unsupervised clustering of the first several hundred, the brains all cluster together for gene expression, and the subregions of the brain cluster together. So it's looking pretty good, but it's always going to be a. I should be quiet and let.
I think your timing is just perfect.
Okay.
So it's really a pleasure to be back home, as Ruth said, to be back at Hopkins and seeing so many old friends and people I know who have been attracted to come to the NIH. I mean, I'm to Johns Hopkins, and I'm at NIH, and it's great working collaboratively with so many of you. Since we bothered with slides, I'll put up the obligatory disclaimer slide. This is just me talking, not the federal government agencies that employ me, and I have no financial conflicts of interest to disclose, being a good ethicist and all. I think that the Nobel Committee actually did us a service and kept us from having to put too many slides on iPS cells in our presentations. You may be aware that they awarded Shinya Yamanaka the Nobel Prize. So he was one of the recipients of the Nobel Prize this year in medicine for his discovery of the ability to create induced pluripotent stem cells first from mice fibroblasts, and then he sort of tied for the human discovery. And there's been quite a lot of excitement about iPS cells in the ethical literature, claims that because this obviates the need to source stem cells from blastocysts, we don't have to destroy embryos anymore, and isn't that ethically superior? And also a lot of excitement about the possibilities of iPSCs for autologous therapy. The idea you could take my cells, convert them into something I need, and then put them back in me might help with the problem of immune rejection. So a lot of real excitement about the regenerative medicine possibilities. There are some in the bioethics community who thought that maybe jumping to the conclusion that iPS cells are free from ethical concern was a little bit premature. And so some of us at the NIH have been working together to think through what some of the possibly more subtle ethical issues are around the use of iPS cells in research, the fact that they don't necessarily completely obviate the need for using human embryonic stem cells, which are still really the gold standard in research to which iPS cells are compared. So if you really look at the issue closely, there are some issues related to complicity if you're in favor of iPS cells. But I'm actually going to set those aside and look at an issue that we've been paying particularly close attention to, which is the issue of informed consent. So there's a relatively new Center for Regenerative Medicine. That's what CRM stands for at the NIH. Mahendra Rao was appointed in the last year as the director. And it was in the conversations between the gtex project and CRM that they were exploring the possibility of creating iPS lines from all of these valuable tissues that are being collected in gtex. And they requested an ethics consult to explore a few specific questions related to the next of kin authorization process. First, a sort of broad question: how much information would family members, the next of kin decision makers, need about iPS cells in order to make inform decisions about the use of their deceased relative specimens in this line of research. And then a more specific question, given that there was already an existing authorization form that was created fairly carefully, there was the question whether this form is sufficient, even though it doesn't happen to mention anything about stem cells or iPS cells explicitly. And I think a question that is underlying both of these really is simply whether the creation of iPS cell lines is any different from other broad research uses of biospecimens that we're used to talking about for genomics and for the creation of immortalized cell lines. Is this a new technological development that's so different and so important that we have to pay extra close attention to it in consent forms, or is it really quite similar to what has preceded it? And I think instincts across the NIH, at least initially, varied on this. There were some investigators who were quite surprised that the IRB was even interested in hearing about their proposals to create iPS lines with their specimens, and they saw it as no different. And others who wanted to stop everything until we convened panels and studies and other things to figure out how to do this right. And I think we've arrived somewhere in the middle where the research is proceeding cautiously with some close attention to the consent process. So at the time that this consult request came in for the gtex project, I had already been engaged in a collaboration with the Center for Regenerative Medicine, and it was between them and the Department of Bioethics and the NHGRI, which is where I hold my joint appointments. And this paper was the initial product of that collaboration. We had as our goal to create a template, a consent template that investigators who were prospectively going forward with collecting specimens to create iPS lines for research could use as a model to try to harmonize approaches across institutions. We did a little bit of a content analysis. We collected consent forms that were in existence, and we compared them and saw quite a few differences. And we tried to propose what we thought was a reasonable starting point for entities to consider. Since the research is so collaborative, we wanted to make sure the standards were reasonably compatible between institutions so that specimens could be shared and cell lines could be shared between the institutions. Now, keep in mind the Center for Regenerative Medicine, by definition, is very motivated by the translational aspects of iPSCs. So they understand that basic science is the way to get there, but they're very interested in accelerating the process of translation. So this quote, jointly by Mahendra Rao, who's the director of CRM, and Francis Collins, who you may have heard of, the director of NIH. Although basic science advances occur almost weekly in stem cell research, there is a great desire to explore clinical applications, along with an equally strong desire not to have those applications delayed. And in this particular paper, they enumerated a number of roadblocks to rapid translation, and informed consent appeared somewhere on the list. I'm happy it wasn't the very first item on the list, which is so often where IRB issues land, but number 8, issues of consent related to cell-based manufacture. So one of the goals of our collaboration was to try to anticipate the long-term translational aspects to help ensure that the consent process was as productive as possible, while also protecting autonomy and ensuring that people can make informed decisions about what they were getting into. But it was a way to invite them into a longer-term kind of endeavor where their cells might ultimately become used in cell-based therapies for themselves or for others, and it maintained an ongoing communication, the idea of a sustained interaction where there might be follow-up, and there might be reason to either get future data, to give results, to actually offer participation in a clinical trial. So this is what was behind that initial project. Of course, some of the assumptions that we were working with are quite different than the assumptions in the gtex project. First of all, we were focused on living donors, adult living donors who could actually consent for themselves, and that obviously is one big difference here. Also, scientifically, the aims were much broader. Here, I think in gtex, as Jeff described it, there's a really valuable basic science question, being able to compare the iPS lines that are generated to the original organs within the system that you have access to. And the goals are quite focused on those basic aims. And it's very unlikely that those cell lines will ultimately be used to develop cell-based therapies. And kind of got to thinking about all of the different settings, all of the different sources of specimens that might be used in IPS research. And there are really quite a large number that vary in terms of the original purpose of the collection. They might have been for clinical diagnostic purposes. There's talk of using resources in transfusion medicine reserves, and then there might be explicit consent for research. They vary in terms of different characteristics of the donor, whether the examples exist or are being actively collected, whether or not they're identifiable. And it might be that each of these features is relevant in some way to how we think about the consent process and how, or the authorization process, and how specific it has to be constructed around IPS, the creation of IPS cells and the goals of the research. Now, to also answer that question, just how different IPS cells, it would actually be very valuable to have empirical data on patients and family members and the public's attitudes about IPS cells. And there's surprisingly little empirical data in the literature. The door is wide open to folks who are empirically inclined, as many of you here are at Hopkins. And I understand that there might be some research along these lines underway. But I came across in the literature one very large study that was done in Japan, which has a whole media program for education and promoting IPS research, since that's where Shinya Yamanaka hails from. I think it's connected to the fact that the discovery occurred there. So in this internet-based survey of people recruited through a particular newspaper there, they found that a substantial number of people who completed the survey, although they were positive about the idea of IPS research and about the idea of government investment in research, they themselves were hesitant to participate in the research at this time. They were taking a wait-and-see approach and wanted to see more research progress before they would get engaged in research themselves. Nearly a third were concerned about the production of germ cells, of gametes from IPS cells, and thought this should be banned outright. And a significant number received their primary information on IPS cells from the media, from television and from newspapers. And I think these details indicate that there's a role for the informed consent process to play, educating people about the research, if they're being invited to participate in it, if their background information is coming solely from the media. Okay. So let's examine for a moment what it is about IPS cells. What features do they have that might be relevant to the consent process? And this comes from a paper that Bernie Lowe and his colleagues wrote that analyzed this a few years back, that I think still stands as one of the best analyses of this. So IPS cells have the characteristic that they're immortal. They can be grown indefinitely. They can be shared broadly. They also have the feature of being pluripotent, so they can be converted. They can be sourced from almost any tissue in the human body, and they can give rise to almost any tissue in the human body, including gametes. They're likely to be used broadly in a number of different areas of research, including studies with human-animal chimeras within the limits of what regulations allow in that area. They're likely to be used for the drug development. They're likely to be subjected to genetic and genomic analysis, possibly in regenerative medicine, possibly in reproductive and human development types of research, and probably a number of uses that we haven't even begun to anticipate just yet. And this group concluded that, in general, it would behoove consent forms that involve the use of biospecimens in general to cover all of these different themes, since any specimens that are collected today have the potential to be used in any of these ways. And if these were covered in a consent form, then it would be, you could assume that IPS research was covered in that consent form. So now I'm going to turn to the specific language of the gtex authorization form that they already had in place, that was approved. It was developed, I think, in a process that included engagement with the community advisory board that had been put together for that study. And we see that a lot of those domains that I just mentioned are actually covered in the authorization language. So it covers, in several different ways, the idea of broad, open-ended future use, that the type of their samples and data will be saved for many years. They'll be shared with researchers around the world, used in many kinds of medical research, no limitations on the types of research mentioned. It talks about immortalized cell lines, the idea that the samples will be treated so they can grow indefinitely and be used for many years. It talks about genetic analysis and actually provides a clear definition of what genes are and what this means. It gets into the idea of how research results from these specimens will be handled, essentially that they won't be disclosed to family members, but they're upfront about this. They mentioned the idea of translational research, that new drugs, tests, and other products might be developed, and it acknowledges there's a commercial aspect to that. If this happens, if these products are developed, your family won't see any of the profits from that research. And it even talks about the ability of family members to change their mind and what some of the limits on their ability to withdraw from the research are. For example, the family members can't stop samples and information from being used in studies that have already started. They could authorize, or they could discontinue the use in future studies, but for anything where data had already been used in an analysis, that couldn't be withdrawn. And so going through the language of this authorization form, the group that had convened for this consult felt that, first of all, the form was very well written in general. It was very clear. It was written at a nice reading level. It was succinct, and it got to all of these points. The group also considered some other issues as well. The fact that these particular family members were in varying stages of bereavement. They had gone through the process of authorizing consent, possibly for organ donation as well as for this research, but they had sort of moved on from there. And so we talked about whether it would be burdensome to sort of reopen the conversation and ask them about a very specific nuance of the research that wasn't mentioned explicitly, and whether the burden of doing that was balanced by the benefit of them being fully informed about this new research purpose. And there was a feeling that the burden could be quite substantial and worrisome and not worth re-engaging people in this way. There was also a message that had been received in the beginning for this study to have the consent form be as succinct as possible. This was a value that the Community Advisory Board, I think, brought to the table. And so expanding it, including additional language to describe this particular use, especially given that there might be future uses that hadn't yet been anticipated, was not something that they wanted to undertake lightly, because it would necessarily expand the form and make it lengthier. And there was also some discussion about what this might mean in terms of the future possible scientific applications. So if the consent form didn't leave open the possibility of recontacting family members, of discussing iPS cells explicitly, it might limit the ability to use these cells in therapeutic applications because of certain FDA requirements and other things. But there really, at the end of the day, the group realized that there was almost no way that these particular cell lines were going to be used in therapeutic applications, and that there were sufficient plans in place for the MTA's material transfer agreements and other governance structures that would prevent future uses that were not covered by the consent form, like the creation of gametes for reproductive research, other kinds of applications. So the repository where these specimens are going to be kept has a very strict set of rules about under what conditions they will share the specimens with other researchers, and these uses are precluded. So any uses that family members might have been concerned about, for example, things that might have been relevant to their decision because they might have objected, those were likely to be addressed by the MTA and the governance structure in place. And so that reduced the sense that they had to be described explicitly in the consent form. So the recommendations that we came up with were focused on the sufficiency of the existing language. It seemed to us that the current authorization language was sufficient and that it was unnecessary, and in fact it might be problematic to recontact family members to give them just a little more information about iPS research in this particular new area. And this was true not only for the specimens that had already been collected, but for this scaling up effort after the pilot, the idea of contacting or recruiting large numbers of new participants in gtex. that revising the consent form didn't seem like something that was necessary either, that there was enough content in the consent form to cover the sort of ethically relevant features of IPS cells, and lengthening the form to talk about that specific use was as unnecessary. But we did think that it would be a good idea to float these recommendations by the Community Advisory Board, which does include some actual next of kin decision makers who have authorized the use of their deceased relative specimens in this research, to see if they agreed with these recommendations. And we also thought it might be helpful to generate some general educational materials to make them available to family members who might want to know more about what is happening with their loved one's specimens, including IPS cell research, which is consistent with the kinds of materials that are already available. So the proposal, we had a conversation with Laura Siminoff about what the conversation with the Community Advisory Board might look like, whether they feel that consent for broad research purposes covers IPS. It would require educating the Community Advisory Board about what IPS cells are. Are there any conditions, IPS research, something else that would justify the contact of family members, next of kin surrogates, and then to consult them too about the development of educational materials that might be made available to subjects. All right, and just a parting note, these questions about, so we started in our collaboration looking at prospective consent for IPS research, unrelated to this consult that I've been talking about, to come up with a template, sort of the idealized scenario when you have a chance to sit down, talk to somebody, and tell them everything you want to know, they might want to know about IPS research. But there are so many of those other contexts where you don't have those idealized settings, where specimens are already collected, where people change their mind about what's being done with the specimens over time. And so there's a large collaboration going on right now between the International Stem Cell Forum, the California Institute for Regenerative Medicine, some of us at NIH, and also folks up in McGill. Mostly these are funders of stem cell research, but they're looking at issues now related to consent standards for previously collected specimens and when reconsent is required. And this focus that the gtex project has enabled us to look at on deceased donor specimens allows us to sort of bookend the conversation. On this side, we have the prospective consent on this side, and we're going to be looking at everything in the middle in the next year, and hopefully we'll be generating some guidelines related to that as well. Okay, so now I think the floor is open for questions.
Maybe, I just wanted to make one tiny clarification. The Community Advisory Board actually had a form before the consent forms were developed.
Oh, okay.
But so it was working, the NIH folks and NCI and HRI Joy Boyer, and it was Thompson before she retired, and Nicole Lockhart. And then the University of Pennsylvania and the Roswell Park IRBs actually were the ones that approved it. But the organ procurement organizations themselves are also, we're very engaged in this, and really, they're, in my estimation, very understandably sort of protective, if you will, of their families. They go to great lengths to establish a setting where they can develop trust, and so the last thing they wanted was for something to go wrong with this part of a consent. And
thank you. I also forgot to mention, you had told me that the Community Advisory Board we don't yet know what they said about this process and the idea of broad consent. I think the engagement has occurred, but we're waiting to hear back from them. So I gathered it hasn't gone forward yet, and we're still waiting to hear, at least for the ramping up part of it. To hear back. So sorry, Holly.
So I wanted to follow up on maybe some of the logistics. So the gtex will exist, and there'll be some subset of fibroblast cells where the IPS question might be relevant. Is the material transfer agreement, is that the way that access to the samples will be sort of brokered? Or how will, right, so what, if I want to borrow some of your fibroblast cells to make gametes, someone's going to say no. And I'm wondering who might be the person who's going to say no in that setting?
Go ahead. I believe it's the specimen repository. It's where the cell lines are physically being banked, and they have negotiated through a governance and oversight structure that includes an MTA that researchers who request the cell lines have to agree to, that limits their ability. I think that is administered through the
So
repository.
those restrictions, I assume, would come through the MTA and the repository. We don't have any cell lines,
Right.
so
It's a future question, but that's, once it's all up and running, there'll be a way, an external body or not external, but a body that's going to be able to say, here's what the standard MTA ought to look like. We're going to make sure these things aren't, pursued with the cells, and by signing it, they're agreeing to not do particular things.
And it's based on a fairly standard MTA that the Center for Regenerative Medicine at the NIH has worked out with our tech transfer folks. So I think it's a model that they'll be adopting for this collection that's also being used for some other stem cell line collections as well.
access to the tissue repository more generally, we're still establishing that setup. The cell lines are a little bit different because they are a renewable resource. So we put a bit more focus on scientific review and just sort of programmatic resource review of the limiting samples. But,
right, because there's the finite and the infinite,
Yeah,
quote
that's
unquote.
right.
Yeah. Thanks.
Can I just follow up?
Oh, sorry. Yep.
I'm sorry.
No, go ahead.
no follow-ups allowed, sorry.
Do you guys have a sense of whether there'll be a limitation on those you sharing with others? Does that make sense?
That's right. So they would be precluded from passing those cell lines on. I mean, I think that's fairly standard in MTA, but both for cell lines or for the residual sort of frozen or fixed tissues.
Yep.
Thank you for your presentation, and I may have missed this right at the beginning of the presentation, but I was wondering if you could share a little bit on the demographics of participants who consented to provide samples. I'm just wondering how this may contribute to either the elimination or exacerbation of health disparities that we see among the underserved and underrepresented population.
Right. Yeah, the data is still rolling in, so it's sort of a moving target. But the inclusion criteria are very broad. We take donors of any racial, ethnic group. They have to be between 21 and 70, and except for some infectious sort of limitations, anybody can participate. The collection sites are essentially in Virginia Beach, Philadelphia, and Buffalo. And I think of the first 150 donors or so, 11 or 12% were African American, very few Hispanics, a couple percent Asian American. So it's not dramatically different than U.S. demographics, except probably for Hispanic individuals. We'll probably be adding at least one more sort of collection sub-site, and we're hoping that we could improve the participation or increase the representation of that group. But so we basically take sort of all comers, if you will. We don't exclude, or there any kind of restrictions based on that. But is that? So?
And is your community advisory board reflective of the U.S. population as well?
You know, I don't know for sure.
It's a great question. I'm not sure.
Yeah, so that's out of Richmond, and I know it has some families. So they and Laura's working mostly with she's working with NDRI, the two NDRI subsites, so Virginia Beach and Philadelphia. I honestly don't know. Yeah.
Thank you.
There's a question back there.
So I'm working with the informed consent and governance work group of the return of results and Caesar Consortium, and you're echoing conversations that I have sat in on many. And one of the things that we're struggling with in the context of whole genome sequencing research is what the appropriate balance should be between informed consent and governance, and to what extent strengthening governance can address some of the limitations of the informed consent process. And so I have two questions. So one is, what or issues related to governance included in the informed consent document to try to bolster trust in the system?
That's a good question. So I have the consent form sitting right over there, and I'm tempted to go take a look to get the answer to your
I
last
think there's
question.
little or nothing about governance per se in the consent forms.
But I think, I think there is a role for governance and there's a role for consent. I think the group wasn't thinking that governance makes up for shortcomings of the consent process, but that there are certain aspects of the research that are properly situated within governance. So you need to have an excellent plan for the protection of confidentiality, and that's managed primarily in governance, and a consent form mentions just enough about that plan to reassure people that their stuff is being handled in a confidential manner, but it doesn't get into the nitty-gritty details of a coding. That's just one example. I think that because of the scientific aims of this endeavor, and because the governance related to what other researchers can actually do, because there's controlled access and limitations on what other researchers can do with these specimens, I think, I might go against what I just said, but I think that helps to uncomplicate the consent process. We're not asking people to wade through difficult decisions about research that they might have some concerns about. The idea that their deceased relatives, loved ones, cells might be used to make gametes that would be used in reproductive research. I mean, that would get very complicated very fast. But since that's not happening at all as part of the governance, I think that helps to uncomplicate the consent process. But I think there's a legitimate line of research in reproductive health that could be done with stem cells, and I think one could get consent to do that, hopefully from adults who are able to make these decisions on their own behalf, maybe as a starting point. I think that just the consent process looks very different because the aims are very different, and then there will be a different kind of corresponding governance structure. But I don't think we're using one to fix the other. I think they both come out of what the actual aims of the science are and what the plan is to carry it out that corresponds to that. Does that address your question? I've been seeing them. I think you were next.
I guess sort of a corollary to that, you said there were 25 people that declined? So far,
20 people that have been interviewed.
Oh, that have been interviewed. I mean, do you have a sense as to why they?
Not really. I mean, we have a meeting coming up, an in-person meeting in December, so hopefully we'll hear a little bit. Again, we're just so early on. I think in at least a couple of cases, if I recall, I mean, people were sort of at their limit. I mean, if it's a tissue donor, they've sat through an hour and a half or two telephone interview, a reproductive and social and behavioral history. I think they're just, sort of, I've had enough. I don't want to hear about anything more. We haven't really seen a formal analysis or any sort of frequency distribution of those. I think Gail was next, and then. Sorry.
So thank you also for a great presentation. I'm struggling to try to understand the science well enough to think of the potential clinical utility of some of the information you might derive for family members. And the reason I'm asking about that is that I know we focus a lot on confidentiality and ethics as the thing that people are most concerned about, but I mean, Dave knows better than I do. A lot of the research that we've done in other biobanking contexts, people are at least as concerned about getting pertinent information for themselves and their own use. So is there anything about these iPSC cells that would be useful to the people who are consenting, and is that part of the consent discussion?
So it's an interesting question. I think, well, the International Stem Cell Forum, its last project tackled the question of disclosure of research results from stem cell research that might come out of it to individuals and maybe by extension to family members. And they actually put out a position paper that argues, unlike where a lot of the conversation has been going in bioethics, that there's sort of an obligation to disclose genomic. Clinically useful. There's so much uncertainty about the accuracy of the information coming out of stem cell research. There's the mutation rate, maybe that's something you can help me speak to a little more scientifically, but there's concerns about the genomic information being accurate and a need for confirmation at a level that is beyond what other types of genomic research are faced with. So that body suggested that given the current state of the technology, it's going to be unlikely that there's going to be results that are going to be accurate enough to feel that we should be disclosing them to individuals, and then even more so by extension to family members at this time. They were dealing with the tricky question of information coming from research on embryos derived from two people, and the uncertainty about whose genetic information, which family line you were even talking about. So there were some additional complications there that I think I'm getting out of the realm of my ability to describe. But I think part of the goals of this research will be to look at how the homology between the cell lines and the original tissue, and to see how, and to answer some basic science questions that might ultimately lead to having better information that might be disclosable. But I think we're far away from that scientifically at this point. Does that sound
I
right to
would
you?
agree. I mean, I think my impression is that the chance of there being an individually clinically relevant thing come out of the IPS would be much less than what one might argue would come out of just the standard research that we might do.
Well, I'm just making this up. You mentioned taking a brain. Let's say somebody died of Alzheimer's disease, and let's say Alzheimer's disease runs in the family, and and you discover, I'm making this up now, but you discover something about the iPSC cells that may be more or less likely to become cells. I mean, I don't even have the language for it, but to exacerbate or to mitigate the risk of developing Alzheimer's disease. And could those cells be actually transplanted into at-risk family members?
Well, I mean, sort of no, based on the MTA and just sort of FDA kind of regulations. I mean, these cells would never have the potential to be put back into another human being.
Okay.
I mean, some of the knowledge, I mean, you're right, although I would, even so, I would think what we would learn from studying the gene expression in the actual brain tissue that we study would be much greater than something down the line extrapolating to an iPS cell. But
But fundamentally, these tissues aren't being collected with the GMP and all the other standards
that's
that FDA requires for transplant. Unlike the organs that are being harvested, I think they're not collecting these with the intention. So I think there's just regulatory issues too that would guard against what you're talking about. Jeff?
So just to follow up on that, because I was thinking the same thing, not so much about the iPS cells, but about the genomic analysis that you're performing on lots of different tissues, which could have meaningful information for the members of those deceased individuals. So it looked like from one of your slides that no information would be returned to members of the family. That's part of the consent process. So this obviously was thought through, and a decision was made that you would not connect information back to family members, correct? And so clinically useful information, incidental findings, all sorts of things that could come up in other kinds of genetic research too. It's not unique to this, of course. So what was the thinking about why to cut the lines?
yeah, I mean, well, it was I guess probably a combination of things. Just that sort of, I mean, it would be challenging even in the best of, in other circumstances where you've got a living individual who you could explain in situations where you might return something that we don't have that. The person providing authorization could be a wide range of relatedness or sort of association with the deceased, so that I don't know, I guess it just sort of seemed very fraught with problems to try to explain a setting where we would go back to a family just seemed sort of impractical. And frankly, the organ procurement organization as well, I think, we're also just, I mean, in fact, I mean, you know, one of the sites is just very, very reluctant to allow us to do the LC study because they want to have the contact with their donor families and our, so.
That's an interesting aspect, I think, which makes this different, is because you're accessing tissue through the OPO network, rather than all comers or other ways you'd access genomic information.
So as part of this, thinking back to your question about the decliners that we've interviewed, I mean, it's very hard actually to get a real sort of participation rate because every death that occurs in a medical facility in this country has to be referred to the Organ Procurement Organization for possible organ donation. So, you know, I think, but the OPOs know fairly quickly that many of these are not potential donors. So those don't get into our denominator. It's really the people that have direct contact with an OPO person to talk about the organ donation process. And then typically it's people who do go through with organ and tissue donation. And they're about, I mean, I think it's running approximately 3/4 of people then also agree to participate in gtex. So, which still is an ultra small fraction of all the initial referrals to the OPO. But
That protectiveness sounds surprising. Like if these people are interested enough to want to participate, you know, why the reluctance to engage them and involve them? Is that common? I don't know
Well,
the field.
I mean, the subjects are dead, right? So part of it is saying, you know, how would you explain it? You can't explain the use and the intended use to someone who's dead. It would be very different if I was talking to Sarah about the donation of her information, right? I can't, if Sarah's not alive, I can't.
But the family
get her
member has
information.
has given permission. So I mean, you think that they have some interest in research, in seeing something useful come of this. You know, like
Well,
they're.
something useful is going to come of it. What you're saying is what if clinically relevant information?
Oh no, I don't necessarily mean for clinical relevant. I just meant for any questions that you have, not necessarily, you know, going back to like why that protective stance of, you know, one group doesn't want to let their donors, you know, be contacted, something like that.
maybe I'm sort of overstating that, but it, you know, it's I mean, you know, well, you know, it's a situation where a single sort of unfortunate event can really just tarnish the whole process, and so.
I think, I don't know if I'm overgeneralizing, but we've been trying to engage with the Red Cross to do a study of blood donors to look at sort of motivations to donate blood and how people would feel about repurposing those into a research direction, including IPS, creation of IPS and other things. And they've been very reluctant to help us gain access to their donors because they've got a good thing going. They know how to get people, how to tap into this very focused sense of altruism for one purpose, to have a reserve of, you know, for the blood supply for emergencies. And they're afraid if we start asking questions, they might say, well, is my blood donation actually being used? And it might actually reduce the willingness to donate in the future. Even though there are proposals, policy proposals to do this, they don't want to ask people the question because they're afraid it'll have an impact on donation rates. I don't know if that's what's going on here, but it's been a very interesting thing that when you assume everybody's volunteering and altruistic and it should be easy to recruit people to participate in the survey, they're very nervous about us talking to people.
I'm very nervous to introduce it. It's
Oh,
1:15.
wow, we're halfway.
It's only for a segue because the conversation is really engaging, but we do need to stop. So we thank you both and really for participating.